All right. So welcome everyone to part one of STAT's three-part digital event series, Combating COVID-19, sponsored by Pharma. Thank you for being here. I'm Matt Herper, and in just a bit we'll be joined by Regeneron's George Yancopoulos to talk about how antibody drugs can target the novel coronavirus. We've got a lot to cover, but first, a few quick housekeeping items. At the bottom of your screen, you should see a few icons. There's a chat icon and a Q&A icon. Use the chat feature to chat amongst yourselves or to send a message directly to the STAT team. If you're having technical difficulties, you should tell us in the chat. Do not put your questions for George in the chat. Your questions should go in the Q&A tab. So if you do have a question, please type it into the Q&A feature and we will answer it live. We will not be using the raise hand feature during today's webinar. So again, type your questions into the Q&A and please do, we really want them. We want this to be participatory. So thanks again to all of you for being here. Before we get started, I'd like to first welcome Stephen J. Ubel, President and Chief Executive of Pharma, to introduce today's session. Take it away, Steve.
Thanks so much, Matt. We at Pharma are delighted to partner with you and STAT on a three-part digital series where you'll hear from our company's leading scientists and experts on what they're doing to combat the virus. As you know, the companies are literally working around the clock, really on 4 different fronts. We're working with other life sciences companies on diagnostic testing. In fact, one of our member companies tapped their PCR capacity to start a drive-through testing center in Indianapolis. Our companies are screening their vast global libraries of existing medicines and making those treatments available to patients right now in the context of clinical trials, as well as compassionate and other emergency uses. At last count, there are over 284 such trials underway. They fall broadly into a few categories: antimalarials, antivirals, and immunologic agents, which I'm sure we'll hear more about from George today. And they're developing new treatments, whether it's plasma-based antibody approaches or biologic-based antibody approaches that can all serve as an effective bridge until we get a vaccine. And at last count, there are over 50 vaccine programs underway, with a few in human trials. And that's important because we're going to need a lot of shots on goal if we're going to be successful. I'm cautiously optimistic that the industry is going to deliver on all of these fronts, really for 3 key reasons. One is the companies are built for this, with decades of experience with similar viruses like SARS, MERS, HIV, and others, and are tapping that deep scientific knowledge. The companies have also made significant investments in recent years in key technologies that are shortening the amount of time it takes to decode a virus and get lead candidates into trials. So you may recall a decade ago, it took almost 2 years to get the first vaccine candidates into human trials for SARS. And it literally only took a month or 2 to get lead candidates into human trials with COVID-19. Our industry also has unique manufacturing capacity, and our companies are already ramping up that manufacturing capacity at risk, despite not knowing whether their candidates will succeed. And there's unprecedented sharing of information among the companies going on in terms of not only clinical trials, manufacturing issues. We have a regular dialogue with FDA on both treatments and vaccines, as well as seamless interaction with NIH. And maybe I'll just close by saying our companies are keenly aware of the moment we're in, what's at stake in terms of affordability. The industry's track record in previous pandemics is strong. I don't anticipate that affordability is going to be an issue. So I'll close there, Matt. Again, we're delighted to partner with STAT on this series, and I look forward to hearing from George.
Thank you, Steve. And now, a man who needs no introduction, George Yankopoulos, co-founder, president, and chief scientific officer of Regeneron. George was recruited by Regeneron CEO Leonard Schleifer more than 30 years ago, and he's led the development of the company's platforms for creating antibody drugs, including Eylea for macular degeneration, and the potential coronavirus treatments we're about to be talking about. Thanks for joining us, George. And I want to remind all of you to keep submitting questions through the Q&A feature at the bottom of your screen. I will turn to those as soon as I can, as soon as we get some basic information about the studies going. George, welcome.
Glad to be here. That was a great introduction by Stephen about how our industry is trying to really come together in these times when we're literally all under attack. We're all in this together. And hopefully, like he said, we can deliver and we can really make a difference to change the course of this crisis.
So you've been approaching this on two tracks, really. Regeneron has two programs that are targeting the virus. One, which we're going to get to in a second, is the antiviral antibodies. But let's talk about Kevzara a little bit first, because you were fast to start these trials, and it's one of the first things that we have that may come down the pike as being helpful are these IL-6 drugs to treat acute respiratory distress syndrome, like when people get really sick and they're in the hospital and their lungs are inflamed. The excitement of this was a small study with another drug with 21 ventilated patients, where they reported that their body temperature dramatically returned to normal. You and your partner Sanofi are running two trials. How quickly, explain, I've heard a lot of questions about the design of those and how quickly we're going to get results. I think because it's going to be a two-step process. So I thought it'd be helpful to walk people through that.
Right, just to take one step back, as Stephen said, I think we're all agreed that hopefully the final answer here is going to be an effective and broadly available vaccine. And what we're talking about, what you're talking about are, Stephen referred to them as bridge approaches or therapies that in the short term can really impact things. And as you were just saying, unfortunately, one of the devastating aspects of this disease is it's been filling up hospitals with these acute care patients needing ICUs, needing ventilators, and so forth. And what you're referring to is the exciting but very anecdotal small series of data that came out of China that suggested for these seriously ill patients, they may be suffering, their lung disease may not be necessarily due to the virus per se itself, but to too much inflammation as a response to the virus. Much as we see in autoinflammatory diseases like rheumatoid arthritis, for example, and others, where it's the body's response, excess inflammation that causes the problem, in this case, filling the lungs with white blood cells and fluid that makes it hard to breathe. And as you said, because of these results from China and because of this impending need that we saw coming, we were talking with people in Asia. We were talking, unfortunately, just as the epidemic was breaking in Italy, and we were hearing from the front lines in those places what was happening in the hospitals. And it was great to have the hope of this short China experience, but I think many of us in America, seeing the impending doom, felt that it was so critical to get really well-controlled data, because so many times you have a little hope, people are desperate, they're willing to try anything. I'm sure thousands and thousands of people in the United States have already been treated with these IL-6 receptor antibodies. Remember, there's two approved ones. There's the one from Roche Genentech, Actimra, and there's ours, which is known as Kevzara, that we partnered with Sanofi, that are both approved primarily for rheumatoid arthritis. So there was a lot of, obviously, a lot of desperation to use these drugs. But really from the beginning, we formed a great partnership with all the relevant and interested parties. I mean, frankly, starting with the federal government, starting with our state government. We were on the phone with Governor Cuomo. He said, hey, we see what's happening in Italy. We're desperately afraid this is going to be happening in New York. And this was very prescient because this was a few weeks ago when I don't think any of us really knew the horrible devastation we were going to have. We were on, we were having calls with the FDA. They're saying, we got to find out whether these drugs work. So I think in an unprecedented way, state governments, federal governments, the FDA, BARDA, which is the rapid defense arm of the Department for Health and Human Services, led by Rick Bright, and all the great medical institutions, initially in New York, but elsewhere, said, we got to really know, we got to test this. So in an unprecedented fashion, within, I would say, a week or two, we started these first controlled studies, first in New York and now nationwide. We are probably a couple of weeks to at most a month or so away from getting controlled data answer to really understand whether this shot really makes a difference in a controlled way in patients. And I think we all need to know, because we're all so desperate. We want these things to work, but we also need to be sure, and we need to know if they do work, what is their impact? And that's what we're trying to figure out and we're trying to do. And that's why we work so hard to get the study up and going.
And this is a two-step, this is an adaptive design. So that data we're going to get in weeks or months is going to be what? It's going to be on things like fevers, right?
Well, yes, it's an adaptive design that was actually recommended to us by the FDA. They said, look, we don't know enough about the disease to really even model the study. So it was Janet Woodcock, the head of the drug division of the FDA, and we were talking with her. We were on the phone with her and the commissioner of the FDA, Stephen Hahn, at midnight on a weekend. And they were all saying, well, we got to do what's right here. We got to figure out how to, as quickly as possible, get into a study. We're going to forego a lot of the normal processes. And we encourage you to do an adaptive design where, since we don't know what the real endpoints are, should the endpoint be time on a ventilator, getting off of an ventilator, total death rate, getting onto a ventilator, oxygenation, all these, who knew? So they said, design the study so you do a relatively large phase 2 portion. We look at the data together. We determine, is there anything going on here? And can we then design a definitive endpoint analysis? And then we will be able to test it formally after getting, hopefully, some real signal in the phase 2 part, test it in the phase 3. But keep the study going, keep enrolling, so that you don't do what you normally do. You stop, you enroll your phase 2, you take a while, you look at things, months go by, then you initiate a phase 3. They said, keep the study going. And then once we do the analysis for phase 2, you will almost be able to almost seamlessly start looking at the phase 3 data, or maybe even have enough patients for the phase 3 analysis. And, it's sort of bad news, good news. I have to admit, I was talking to people in Italy, and, we all theoretically understood the risks and the possibilities, and this is why we were working so hard. But I can't believe the devastation we're facing here in New York at the epicenter. And the bad news is so many, tragically, people have already died and are in hospitals and on ventilators. And that's allowed us to enroll our studies so much faster than we were anticipating. And it's unfortunate, but hopefully it means that we'll get some definitive answers here sooner rather than later. We'll know, is it working, or at least do we have some endpoints? And then we can almost immediately, I think, start getting answers from the phase 3. So I think, yes, we're a couple weeks away, hopefully, from knowing about the first part, the phase 2, and then just a few more weeks to maybe, because we will have been enrolling the phase 3, and we're getting, unfortunately, so many patients that we will literally be able to, if we have anything in the phase 2, we'll be able to unblind the phase 3 portion and apply the endpoint analysis that we, together with the FDA, will try to come up with. But we should all know that, biology is very difficult when you have, these early stages in the disease, and you have these anecdotes. There's always anecdotes. It's always, the odds are always against you. But of course, we're all praying, because we're all in this together, that we're going to see a strong signal here.
And you'll be able to do the phase 3 analysis on patients who are already enrolled once you've looked at that phase 2.
Exactly.
So that's why this could be
much faster than normal.
So this could be months, not a month or two, not fall.
Exactly. No, that's exactly the hope here, because normally, as we said, you do phase 2, you wait, you do an analysis, you wait for a month or two to talk to the FDA and so forth. Everybody's working hard to be looking at this hand in hand in a partnership in real time. We will be enrolling the phase 3. Of course, we'll all be blinded to it. And then if we all agree that there's a signal and we know what the analysis should be, we can literally, depending on how many people we have, almost unblind it immediately and get the definitive answer in the phase 3.
Okay, let's move on to the antibody approach, which the Q&A is filling up with questions on that. But let's start off again by talking about the timelines and the studies. You've said that you've identified a whole bunch of potential antibodies and that you're hoping to start studies by June. Give a quick background on this approach. These are mostly mouse-generated antibodies, although some of them are, you're also looking at the blood of people who've gotten better from COVID-19. Take us through the next couple months as you get toward testing this.
Okay, as Steve said, I mean, we and others have spent decades developing technologies that could be useful here. In our particular case, we've now developed 2 sets of very relevant technologies. The point of a vaccine, you give a vaccine and the goal is to induce a potent immune response against the virus. That means a potent antibody response. That is high levels of potently binding and neutralizing antibodies. That's what you want out of a vaccine. The reason why there's, as Stephen said, many companies working on vaccines using many approaches is it's going to take significant effort, unless we get lucky, to get an approach that really in a high percentage of people safely raises high enough antibody neutralizing levels. Our approaches allow us to get the end product of the vaccine, which are these highly potent neutralizing antibodies, to get them outside of the body, grow them up to high levels, purify them, only the best antibodies, and inject them back in, essentially delivering to the patient exactly what a vaccine is intended to do, but hopefully more potently, more safely, much more quickly, to allow what is called a passive vaccine approach and even a treatment approach. This is exactly the approach that we used for Ebola, which remarkably, because that's in some ways even a more devastating disease if you get it, with a much, much higher death rate. As you may recall from the studies that were carried out with our antibody cocktail by the World Health Organization, in patients who were earlier in the stage of the disease, you could save 90% of them. And you could save about half the people at the latest stages of the disease, which is really remarkable. We're hoping that the same approach will yield same sort of results here. So what is the approach? As you said, there's 2 approaches that we undertake and we've developed over the decades in parallel. One is we don't need human survivors. We have these mice that essentially we genetically engineer to have a fully human immune system. So when you challenge them with the virus or viral components, they generate a potent antiviral response. It's fully human. We can go in there, we can clone it out, purify it, test it, get the best antibodies out, and then grow them up, eventually purify them and give them back into humans as a treatment. In parallel, in this very related technology, we can also go to convalescent, recovering, or human survivors, and using essentially parallel strategies and approaches, get the best antibodies out of those patients. And so I believe we're one of the only, if not the only company, that has these 2 approaches going on side by side, hand in hand. And what I believe that we announced weeks ago now is that we had initiated, these are all under the leadership of an incredible young scientist here at Regeneron, Christos Kyratsous, who works with many of the integrated capabilities and technologies that allow him to do this at Regeneron. He's the one who actually successfully led the effort for Ebola. And a few months ago, we started these efforts, and we announced somewhere around a month ago now that we had successfully gotten thousands of antibodies out of both the genetically humanized mouse approach and out of human survivors. And we have a variety of high throughput automated screening approaches that we're able to rapidly go through these thousands of antibodies and get the best of the antibodies. And, looking at the antibodies from both approaches, we had hundreds of candidates of antibodies that were terrific at blocking and neutralizing the antibody. And so what we've now selected is a series of leads to go in the scale-up process. It takes quite a bit of work to go from, once you have the antibodies identified and cloned and in hand, which we believe we have right now, we then have to scale them up and grow them in these large manufacturing bioreactors. So we've done everything to get to that point, and we're actually on target in a week to start the process of growing these up in these large-scale bioreactors. And so, this is biology. A lot of things can go wrong. There's no guarantees that these things will work, but we are very excited. This very targeted approach, generating very potent, highly neutralizing antibodies, just like what we did for Ebola. And we, by the way, have done the same thing with MERS and so forth, that this really has a chance of working. And we are on schedule now in June to be putting these antibodies in human trials. Like I said, there's always risks. This is biology. It's not straightforward. Things can go wrong. There's no guarantees that these will work, but I think that these are a real legitimate science, target-based shot on goal here that I think that we are really hoping can make a difference. And it can make a difference both for prophylaxis, that is protecting people who have yet to be infected, people like critical care workers, healthcare workers, people at the front lines, first responders and so forth, or people with high risk factors and so forth. So we can protect uninfected people, but we also hope, just like in Ebola, that it'll actually be able to treat people in the early stages of the infection, that is, people who have symptoms but have not really progressed to severe lung distress and so forth. If anything, based on the Ebola experience, it should work even better in those people and prevent progression. But we also hope that it'll work in the same population that we're testing the Kevzara, the anti-IL-6 receptor antibody. Those that are already in the hospital, they're already having trouble breathing, they may already be on ventilators. Can we actually help there as well? And so I think there's a lot of reason to have hope. Like I said, no guarantees, things can go wrong, but we've generated really very potent antibodies here, very effective at neutralizing the antibody. They're in the process of being scaled up and purified, and we are on track in June to be starting 3 sets of clinical trials like I just described.
Okay, really a whole bunch of questions coming through that boil down to all sorts of reasons that antibodies might not work. As you mentioned, biology being difficult. I want to ask one that I think is representative, but is also an interesting take on it, from Nils Lomberg, who knows antibodies. Can Dr. Yancopoulos comment on a recent report by Wolf et al. That viral titers do not appear to drop dramatically on seroconversion? So basically that some people's antibodies may not be as, that the protection may not be as much as we'd expect there. How have you been thinking about this?
So we're saying in patients who are beating the disease.
Mm-hmm.
So by the way, a lot of respect for Niels. He was a pioneer in this field of these genetically humanized human antibody mice, and we were great collaborators a while back. So a lot of respect for Niels. We could actually use him if he wants to come back to the labs here and help us out. But just to understand the question specifically again,
So he's referencing a paper which I haven't seen from Wolf et al. That viral titers do not appear to drop dramatically on seroconversion.
Oh, okay, the virus. I thought he was saying antibody titers.
Yeah.
Okay. We haven't studied that extensively. However, I think that right now, there's always the possibility that this virus is going to behave differently than every other virus, known to man. But I think that we do know that people become viral negative. I actually have friends and relatives who've had coronavirus, and they've gone from being tested for active virus by the PCR assay and so forth, and they convert to negative, and then they get antibodies. Now, it may be that at the time of seroconversion, you may have to have a lot of increase in antibody production and maturation and so forth to eventually shut the virus down. But it seems like eventually you do get patients who are virus negative and they are antibody positive. And we know in our experience with MERS and MERS models that we did and so forth, that we can, if you have highly potent neutralizing antibodies, you can put them in these animal models and they can be both prophylactic, they can completely prevent the disease if you give them early enough in these models, and they can also treat and reverse active disease. So we think that it may be that for whatever reason, maybe this is one of the reasons why there's problems in some patients, that some patients, their immune response starts, but maybe they're not generating potent enough neutralizing antibodies early on in the response to really well control the virus. But that is the whole goal of this recombinant antibody approach, to give the patients what they need to really stop the virus in its track.
Why don't we do this against more viruses? I mean, the only real approved product that we've had in this realm has been Synagis. You talked about a bunch of success stories in emerging diseases, but this isn't usually antibody drugs are against everything but infection, right? They're for cancer, they're for rheumatoid arthritis. Why don't we do this more often?
You know, I think that's a great question. I mean, we've actually been exploring using it in a variety of viral and bacterial infections. I think in general, unfortunately, the world and the industry has deferred a little bit from antibiotic type research, anti-pathogen research, antiviral research, and relied more heavily on vaccines and approaches like that. And I think that this just demonstrates that we have to continue to invest a lot in new approaches, including antibody approaches. And I do think that, as you point out, while Synagis is a great example, where the monoclonal antibody actually replaced convalescent human serum. That was the progression that we're talking about. And convalescent human serum was actually not anywhere near as effective as the actual recombinant antibody. And clearly our experience with Ebola was rather dramatic in the impact that an antibody could have. And I think maybe, yeah, that was a little bit of a shortfall in our industry. But in general, as we've probably all talked about, that there's been less effort against pathogens of all sorts for a variety of reasons than there probably should have been in our industry. And I think we should be using new approaches, and we're seeing it. I mean, unfortunately, this is what pandemics like this are now doing, both vaccine research in terms of whole new approaches for vaccine research, and also whole new approaches against antivirals, including increased implementation of antibody approaches.
So Michelle DeWild asks us, how is the Regeneron approach different than isolating human mAbs directly from convalescent plasma? Is it better?
These are parallel approaches, I think, in my mind. I think the advantages, we're doing both. And one thing that I should say is, I am hopeful, for example, that plasma-derived convalescent human serum products will have some activity. But the concentrations, we already know this just from testing convalescent serum, is that they are only neutralizing at what you call 1 to 100, 1 to 50 titers, okay? Whereas our highly concentrated forms can be diluted out 1 to many, many millions and still be neutralizing. So what convalescent humans give you, we can get recombinantly much, much more powerfully and potently, which is why we're hoping that convalescent serum has some benefit, but creating them and making the recombinant forms will be more potent. The kinds of antibodies you can get from the genetically humanized mice versus the humans are, it turns out, and we probably now have one of the most extensive experiences, they can be very similar. Advantages to the mice is you can do all sorts of what are known adjuvant hyperimmunization approaches, and you can continually sort of beat on the mice immune systems to get more and more and better responses. Whereas with the humans, you sort of get what you get. You're not going to continue to challenge and drive the humans to better and better immune responses.
You can be meaner to the mice.
Yeah, I didn't want to put it that way, but yeah, you can beat on them a lot more. And so that said, though, there is something to be said for human diversity, and we're actually doing this. You can sample, if you wanted to now, unfortunately, hundreds or thousands of humans and hope that some special human might have a jackpot out there. So I think that there's value for having both sets of approaches and being able to compare between them. And right now we have the largest collection of both mouse antibodies and human antibodies against the same target, allowing us, I think, to have the greatest opportunity to choose between these and have selected our best cocktail to be putting forward.
So this was actually the first question we got, and it's one I've been hearing a lot. How fast will the spike protein mutate, and how long will antibodies be effective?
Yeah, these are all great and tough questions. Right now, I would say I would not put spike mutation right now as a problem. And in fact, we have been able to generate all the various or the vast majority of the spike mutant variants that have already appeared and shown and seen that our cocktails are effective against them. That said, there hasn't been any selective pressure put on the spike protein yet. By selective pressure, something targeting the spike protein, like an antibody, to allow it to then have mutants generated that now avoid it. And that's the key reason why we're using a cocktail. Okay, that's what we did for Ebola. So if you only had one antibody in there, then you would be sensitive to possibly a single point mutation, creating a resistant variant that no longer bound that antibody. What we've selected is a cocktail of antibodies that bind differently and block differently to the spike protein. So this is just like, you know, the cocktail combination approaches that were used by HIV, that if you only use one, you can generate and select for mutants, but if you put in the cocktail, it becomes much harder. So that's why we use a cocktail. That's why you use the cocktail for Ebola. That's why we're using a cocktail here. But that said, until we see how the virus responds to the selective pressure of antibodies, of course, you know, it's biology. It's not all entirely predictable. That's going to remain a concern. So it's a concern. We have a cocktail to select against any one mutant antibody to increases the chance that the same virus won't generate multiple mutations on different parts of the spike protein at the same time. History is on our side here, but we won't be able to tell until the experiments are being done.
So we have a question that I think the lay public is asking a lot right now, especially given as we both know how bad things have been, particularly here in New York. Why is it required for there to be a placebo group? Why can't we use historical data as a de facto placebo group, a la the Flatiron Health model? You know, how many people need to be exposed to placebo as we test new therapies?
Well, yeah, once again, I mean, these are great questions. And anytime you design studies, you know, you always have to deal with these questions. Well, I think that with all due respect to all of our abilities to model using machine learning, using all sorts of real world data and so forth, the reason the FDA still demands placebo-controlled studies is that, unfortunately, and you know, we would love to say otherwise, you know, because it would save time, it would save money, and it could save lives if it really worked. Unfortunately, those approaches don't really work really well. That's why the FDA still demands in these settings to have placebo-controlled studies, because until you really do it rigorously, you don't know. And people have historically so many times been fooled with single-arm studies and with real world data comparisons. That said, we work closely with the FDA. And so, for example, in our IL-6 receptor antibody study, our Kevzara study, it's randomized 4 to 1. So 4 out of every 5 patients are getting drug. I have to say, though, that in a weird way, in terms of the common good, we're going to have to now enroll more patients to get enough placebo patients to be able to make the accurate comparison. So by treating less placebos, overall, you have to study more patients, take more time until you have a definitive answer. So it's always hard. I mean, these are hard, sometimes terrible ethical dilemmas that we get into. But right now, unfortunately, nothing exists that's better than doing a rigorous controlled study. The FDA holds you to that. And I, from looking at how many times we and other people have been fooled over the decades, the world needs it to really know whether these things are working.
I mean, you guys experienced it with one of your first drugs. You were on 60 Minutes and they were asking why you weren't giving it. And then the drug turned out to be ineffective.
Yeah, no, I mean, this is what I said. We and others, unfortunately, have been fooled many times in the past. What you're referring to is we had a drug for Lou Gehrig's disease, which is still a horrible disease that we work on. And we have put enormous effort over decades now on it. One of my best friends is suffering from this disease right now. We're very motivated. When we were doing our clinical trials, we worked with the FDA and had a placebo-controlled study. And we were actually accosted on 60 Minutes and said, why are you doing a placebo-controlled study? It's obvious that this drug is working. People are throwing away their canes. They're walking. I mean, this is totally unethical. Well, when the study was unblinded, it turns out that, yeah, people were doing better. People were throwing away their canes. People literally were getting off ventilators because that affects your ability to breathe. It turns out that equal numbers of those patients were on placebo. And this is why it's so critical to be doing these well-controlled studies, because the last thing you want is, out of all the desperation that we have, to be widely using a therapy and maybe to even find out that it's actually harmful because we did not do the controlled studies. So sorry to say, there's still the only rigorous way to really be sure that your treatment actually works and ultimately to save more lives and prevent harm.
A question from Jeff Lazard. Did FDA say anything about the possibility of an accelerated approval based on dramatic phase 2 data? I think this applies to both of the efforts here.
Yeah, I mean, I think, as the FDA always says, rightfully so, and the FDA is a great partner. I have to say, in our more than 30 years of dealing with the FDA, many of them with Janet Woodcock, because she's been there as long as, almost as we've been at Regeneron, they're usually great partners, and they're always trying to do the right thing. They don't slow things down unnecessarily. They're working hand in hand to make sure that you do the right thing, to protect you against your own conflicts, but to get the real answers and as fast as possible, particularly in this situation right here. It's always what they call a review issue. If the data is astounding, I mean, the good news about the phase 2 is that it's a controlled phase 2. We have a placebo group in there. If the results are so striking and so black and white, I am sure that they will take that into consideration. But as they always say, it's a review issue based on the data.
Okay. One thing I've wanted to ask you here is, there are a lot of parallel efforts going on, both in the antibody space, and we also then have the antivirals and the vaccines. How do you feel we as a society should be working? Should we be doing any prioritizing of Regeneron versus anybody else in terms of getting manufacturing up, in terms of supporting these projects? I mean, I think there are hundreds of programs against COVID-19 that have started up. How do you think about that?
Well, I don't know what the right balance is. For sure, there's a value to having several different parallel approaches because, no matter how good things are looking and so forth, things can always go wrong. Like I said, this is biology. So it always makes sense to have several different efforts ongoing, just in case, even if the efforts are very related, something goes wrong that we don't anticipate. That said, at some point, if there really is an effective approach, and if the need is so great, if we need more doses, I mean, we are working hard. Actually, once again, in order to manufacture things, you also need to get FDA approvals for your manufacturing and so forth. So we are working very hard with the FDA, with BARDA, with everybody else, to be able to dedicate and devote our entire upstate New York manufacturing facility, one of the largest manufacturing facilities for biologics in the world, entirely to this effort, whether it be the antiviral antibodies or to the IL-6 receptor antibody effort. But that said, depending on how bad the need is and at the time, the world may need more. And at that point, I really do hope that people start stepping forward, and that might be the appropriate time for people to come together. We're already having discussions with a lot of people, already talking about stepping forward, already talking about maybe offering manufacturing capacity, both in the United States and for the rest of the world, for developing countries and so forth. And I think I'm proud to be part of this ecosystem right now. I mean, when I talk about this ecosystem, the collection of all the people coming together, whether it's academics at major medical centers and the physicians at the front lines there, or whether it be all the people in the biotech and biopharma industries, because right now, when you bring up those issues, I can call up, like I have, the head of Michael Dolson, the head of R&D at Pfizer, and the first question he asks is, well, what can we do to help? Mm-hmm. Okay, maybe you need some manufacturing capacity. Let's talk. Is there an opportunity to help you guys? So I think this is a great time where we can show in unprecedented ways that we can really come together and all do the right thing. And I hope we look back proudly on how we collectively, as a humanity and as a people and as an industry, respond to this. But I see a lot of cooperation. So I think that there's value to some parallel efforts, but I think at some point it will be useful. And I hope, since our antibodies are going to be some of the first that are out there, I hope our antibodies really do work. And then I think that we'll see some unification behind that. And maybe if the need is still there, great, people coming together and saying, hey, yeah, let's help out and let's manufacture these for our country and for the world.
Kind of a little bit of a veering off question, but I'm curious if you have thoughts on it from Nina Shelson, who also, by the way, says that Nils belongs to Canaan and you can't have him. How reliable are antibody assays for measuring vaccine response, and how calibrated for protective immunity? And also, are we confounded when we're measuring immunity by the many, many coronaviruses we're all exposed to, some of which just cause colds, right?
Right. You can't be an expert in everything.
Yep.
Okay. There some of the things that you're asking about, we know a lot about, and we know, things very deeply. Other things we know a lot less about. But I think that what we're seeing is that the human response already to the actual coronavirus, we're not vaccine experts, so we don't know what you need for a positive vaccine. But of course, we are characterizing the human responses to understand how our antibodies might compare to those seen in people who have beaten the coronavirus infection. So we have a different interest, obviously, than the vaccine people. The vaccine people are trying to say what you need in the human response at a minimum to provide protection. We're trying to show that our antibodies, we can use them in a way that goes many, many fold beyond what successful humans have done, so that we can provide it much more potently and immediately than what you generally get. And that is what we're seeing, is that on average, people who have beaten this have what you call relatively low titers, and you can't measure huge amounts of these potent neutralizing antibodies in these people. When we go in and clone them out, we can find them. But it's not like these people have very, very high levels of this. Our goal, which it looks like, hopefully we're on track of getting, is by cloning the most potent ones out, both from our humanized mouse system and from the rare antibodies out of these humans, that we now can achieve much, much higher levels of very potently neutralizing antibodies. And that's our goal, to do much better than the people are doing on their own. And we think that should provide both ample protection against a new infection and maybe even provide a treatment benefit in the severely infected. What we can't speak to is what level of response you would want to get on average in people to get a good vaccine. And I think these are the reasons when Tony Fauci says we could still be, despite all the efforts, we could still be a year or two or more away from a safe and effective vaccine. It's because it's not just inducing the immune response, and inducing the right kind of immune response high enough in enough people. And if you don't do that, then you can get trapped in this unfortunate situation where you could actually be enhancing the viral response and actually getting worse disease. And so these are why you need the experts. And we heard from Stephen, there's probably hundreds of companies working on the vaccines using many different parallel approaches. And I think that's important because I think a lot of these are going to encounter problems, and hopefully at least one is going to succeed, and succeed sooner than later, to address all the issues that you brought up.
I think this is a good transition point for talking about prophylaxis with antibodies. Now, you talked about that there are the two approaches we can use antibodies for. We can use them for treating sick people, various degrees of illness, or we can use them to try and prevent infection. I have to imagine that the preventing infection studies are going to read out later than the ones in really sick people. They're just going to take longer. But walk us through the differences between those two approaches. I think that's a really important thing for people to understand.
Yeah, I mean, that is a great point. And first of all, before we talk about the types of clinical trials, we've done a lot of studies, and what we know, and whether it be from our MERS experience or Ebola experience, the amounts, and this relates to your previous question about what you're seeing in humans right now, to beat the virus, if it's already taken hold, to treat somebody, the later in the disease, you need more and higher antibody levels. To just prevent an infection, you need much less antibody on board. So the prophylactic doses are, I'm just throwing this out there, you know, and I don't want to predict anything, but they're going to be on the order of 10 times lower doses than you need for treatment. And this is because you just don't need as much antibody to block an early, you know, the virus just coming in from a virus that's already taken over and caused a lot of disease and inflammation. As you said, you would think that to see a treatment effect on people who are already sick, that would come faster, as you said, because every patient is somebody who has the viral infection. That said, as I just said, it'll be harder for an antibody to really work in that setting. It did spectacularly in Ebola, so we hope it will spectacularly work here. But if the sickest patients, for example, have already transitioned to a point where it's not really the virus that's causing the problem, it's the body's response to the virus, then the antiviral antibodies may have little benefit in these late-stage patients. So you are exactly right. It'll be faster to get a result in the sick people, and the sicker the people, probably the faster you can figure it out, but you are less likely to impact, and you will need higher antibody levels to impact later in the disease. The prophylaxis approach, it all depends on, you know, and this is something that, of course, we're new to it. I mean, the world is new to the coronavirus epidemic and to the epidemiology of it, how it's spreading, how fast it's spreading. So if, for example, if you were horrifically in a situation where you had a closed environment, like in a nursing home, I tragically right now have a 91-year-old aunt who's trapped in a nursing home where right now there's a coronavirus outbreak, and I just wish I could get them in our antibody today. It's just not ready. But basically, if you had a situation where there was an ongoing outbreak and you were afraid that maybe everybody was going to get infected over the course of the next few weeks, you could do in such a closed environment, you know, a prophylaxis study right away. The problem is it's not necessarily that easy to find a situation where a high percentage of the people in a very short period of time are going to be challenged, as they might be on a cruise ship or on a nursing home in a trapped environment like that. So, as you said, if you end up doing a prophylaxis study sort of just on free-range humans, and if people are social distancing correctly and so forth, it may be a long time to get enough infections to actually see it. This is what we're trying to do. We are working right now. We had a phone call just yesterday with Governor Cuomo's team, the New York State Health Commissioner, Howard Zucker, trying to understand how we can find the best at-risk prophylaxis populations so that we can do it sooner rather than later. Because like you said, if you have to test it in, you know, 100,000 people with a low rate of people getting infected, it will take a long time to get a prophylaxis answer. But it is far more likely to work in prophylaxis than it is in late-stage treatment, just because of the biology of it. and much lower amounts of the drug should work in prophylaxis. So I think it's our obligation, our responsibility to continue to work with everybody, the academic leaders, all the government officials and capabilities they have to understand how to best do a prophylaxis study, an early treatment study, which I think is another one that has an opportunity to work really fast. That is, the people who are sick, mild disease, only a small percentage of them will get serious disease. Can we prevent 100% of those people from getting serious disease? That would be a huge, huge breakthrough, let alone the late stage patients. So we're working 24/7, our clinical team, with all the collaborators we have available to us. Everybody's coming together to try to plan the best, fastest way that we can get the answers in those three settings: prophylaxis, early treatment, late treatment.
Completely different question from Kent Lloyd. Is the Regeneron Genomics Center looking for genetic risk or resilience variants in ACE2, which is a gene that the virus uses to enter human cells, or perhaps other genes from COVID-19 patients? Anything you can tell us?
Yeah, Kent, of course, another valued, respected, longtime, collaborator and fellow geneticist in the business. So, of course, those sorts of ideas would not have escaped him. Yeah, we are actively working on this. We're doing exactly these sorts of things. In fact, I was just before this meeting, I was on a conference call with Eris Barris, the head of our Regeneron Genetics Center, working together with Tom Maniatis, who heads the New York Genome Center, discussing exactly how, and we've done a lot of collaborative projects, with Tom and with others, how we can all get together to get exactly that sort of data faster and better. We have looked, like Kent has said, at existing populations to understand, and we have identified, as he said, ACE2 mutations, other gene mutations, and so forth. The problem is, in our databases right now, the historical rates that we could see haven't given us the power to see a lot, which is why we're now trying to build a whole new database. It's like trying to fly a plane while you're trying to build it. So we are literally working with people like Tom and all the great New York medical institutions and others, people at Northwell and others. Can we sample and do all the genetics and all the transcriptomics and all the proteomics on the people who are in the hospital, the extreme cases, the people, for example, young kids who shouldn't have severe disease? Why are some young kids getting severe disease? To try to understand the genetics there. But we're also trying to establish what you need is a large control population of people who are not getting sick, but are infected and we don't even know about it. So we're trying to do a large sort of public health epidemiologic study. And there, once again, maybe in collaboration with New York State and with Howard Zucker and the New York Health Commissioner. So yes, we are trying to do it. But as you might imagine, I mean, this is a state where we, like everybody else, we're trying to build a plane while we're trying to fly it at the same time. But let us hope that we all collectively, I'm talking about the entire scientific and healthcare industry community, start figuring out how to do some of these things, working together and get some answers. But I think this is an important topic to describe. I think that hopefully as many of us as possible can get through this and survive, and we come out the other side. And the goal should be to come out stronger and to learn lessons from here. And Kent's, I think, question raises a really important opportunity that we have to be thinking about for the future. One thing that I think is just a real shame is, as a nation, we haven't figured out a way to get a truly big data, large integrated healthcare information network system that is accessible to all of us, that we can immediately get this sort of information that Kent is talking about, while simultaneously knowing exactly how many people at any given time in the United States have this particular disease, or have had it in the past, or have been treated with this drug. Then maybe some of the questions about real world, data could actually become useful. We don't have such an integrated, large data healthcare system that's accessible in the United States. Not only one in which we integrate genetics with. It's a little I have to say, it's really unfortunate that we've actually gone outside of the United States. We have a major collaboration with the United Kingdom, with the UK Biobank, to be creating these sorts of databases. I think this is a learning time, that whatever we need to do as a nation to do it, we should be creating such a big data system that doesn't exist. And I think that it would allow us to instantaneously ask the questions that Kent is asking right now. And we won't have to build a whole new airplane to be doing it. I mean, we'll have it built and we'll be just able to, fly it and figure out exactly some of these answers and find out, which drugs that are out there right now might actually already be having an impact, instead of having to do the sort of studies that we're doing with Kevzara and so forth. So it's a real shortfall in our ecosystem. And I think that this is one thing that we're going to have to do when we hopefully all come out of this, is figure out how we can do better to be prepared in the future. And I think that that's an important one, is some sort of big data, large integrated healthcare database that puts in all the possible data that one could have to be asking these sorts of questions and getting immediate answers, coupled with sequence and transcriptomics and all that.
So I wanted to, as a last question, I wanted to ask, there's we're running out of time here, but there are a lot of people who, despite the need here, are looking at the industry and are unhappy with things that companies have done in the past. And whether they're concerned with price gouging or there's just general distrust. I wanted to ask if there's anything you wanted to say to people who are skeptical about the pharmaceutical industry in general, maybe Regeneron in particular. You guys haven't been in as much hot water as some other companies, but is there anything you'd like to say to those people about those issues now?
Well, I am sure that every human, can look back in the past at their experiences, and I'm sure there's things that, every one of us, regrets or hasn't done perfectly or hasn't done right. But I do have to say that I think that it's easy, to have punching bags. It's easy to point fingers. It's easier to criticize. What I see, and this is not only those of us who are in the biopharma industry itself, but scientists in general throughout. I mean, the vast majority are well-meaning people who devote their lives to try to use science to make a difference and address disease. And I think that this is one thing that maybe, once again, we can learn something from this experience, is who the real enemy is. I think we've done a little too much fighting amongst ourselves. I'm talking about in general. I'm talking about as a species. I'm talking vis-a-vis politics. I'm talking about all this sort of stuff. I think that we got to start recognizing who the enemies really are. We face true existential threats, okay? Disease is one of them. I've been talking about this forever. People have been talking about issues like drug pricing and healthcare for all and all this. And I know I told you this, Matt. Okay, I said, while we're worrying about those issues, I'm sorry, all we have to do is look a little in the future. There's going to be just too much disease for us to be able to deal with and to handle. And we have to understand who the real enemy is. And the real enemy is there's too much disease. There's too many environmental problems. There's climate change. And we have to figure out how to come together. And we have to actually, instead of pointing fingers, we have to figure out how to come together and work harder to promote much more investment in innovation. Because we just realized something. Healthcare for all, it's a joke, okay, when there's so much disease. No society can deal with this, and no society can provide healthcare for all if they don't have real, true solutions and cures for diseases. And I think that we have to look at our industry as a critical part of the answer and the solutions. And I see so many great people in our industry. We're all in this together. I have family members. I have friends on ventilators. We're doing this. We're all in this together, and we are killing ourselves 24/7 to try to make a difference here. And we have to start realizing that it's time to put petty differences aside. And, drug pricing, no, it's disease we should have been talking about. We should have been prepared more about how do we invest more in healthcare to incentivize more science, more technology so that we're more prepared. How do we build the sort of database network that I'm talking about? We have to decide where we want to make our investments as a society. I think we should be making the investments not necessarily in, in new apps and new video games and all that. we should be making them in the sort of technology that can fight the biggest problems that are challenging mankind. Disease is one of the top ones, and another one is the environment and climate change. And let's come together and figure out how to do a better job of addressing those instead of going back to partisan politics and finger pointing and dividing ourselves. Let's come together here. Let's show that humanity can rise, okay? You know, like I've said before, we're just like bacteria in an Erlenmeyer flask. The only thing that keeps us from dying from our own disease and pollution is our God-given ability to try to think our way out of that Erlenmeyer flask. And it's time for us to really recognize that, recognize who and what the real threats are, and come together and promote the sort of engagement of mind and spirit to really innovate and invent our way around these truly existential threats.
All right. And we're a little over time, so we'll close out there. Thank you so much for doing this, George. I think this was a great first try at one of these. So thank you for being our first virtual speaker.
And as I've adopted the expression, Godspeed and science speed to all of us. Let's all, get through this together.
Thanks, George. Good luck.
You too.